Benvinguts i benvingudes a l'acte d'investidura d'Emmanuelle Charpentier com a doctora honoris causa per la Universitat Pompeu Fabra. Donem la benvinguda a Laia de Nadal, rectora de la Universitat Pompeu Fabra, i a Marc Güell, padrí de la doctoranda.
És que com vaig amb el pinganillo i ja no sé quin micro he d'utilitzar. Bon dia, sigueu benvingudes i benvinguts a la cerimònia acadèmica d'investidura de la professora Emmanuelle Charpentier com a doctora honoris causa per la Universitat Pompeu Fabra. Us convido realment a gaudir d'aquesta cerimònia que és sobretot un homenatge a la professora Charpentier per la seva magnífica trajectòria acadèmica i intel·lectual i també un tribut a l'excel·lència i a la recerca del coneixement. Gràcies a tothom per ser avui aquí. A continuació, demano a Montserrat Vendrell, presidenta del Consell Social de la Universitat, que pugi i que llegeixi l'acord del Consell de Govern de concessió del títol de doctora honoris causa per la UPF a la doctoranda Emmanuelle Charpentier. Gràcies.
Acord del Consell de Govern de la sessió del 4 de juny del 2024. S'acorda concedir el títol de doctora honoris causa a la doctora Emmanuelle Charpentier a proposta de la rectora en reconeixement a la seva valuosa trajectòria acadèmica i intel·lectual en l'àmbit de la biologia molecular i la genètica, en especial, però també per la seva contribució al desenvolupament i comprensió de la tecnologia CRISPR-Cas9 que ha revolucionat l'edició del genoma i obre la porta a noves teràpies genètiques i estratègies de tractament per a malalties genètiques, càncer o malalties infeccioses, per la seva contribució al debat ètic també i social en aquest àmbit i pel seu intens compromís amb la divulgació científica i l'educació pública. By resolution of the Board of Governors of 4th June 2024, Pompeu Fabra University has awarded Emmanuelle Charpentier the Doctor Honoris Causa degree for her valuable academic and intellectual career in the field of molecular biology and genetics, and in particular for her contribution to the development and understanding of the CRISPR-Cas9 technology, which has revolutionized genome editing and opens the door to new genetic therapies and treatment strategies for genetic diseases, cancer, or infectious diseases, for her contribution to the ethical and social debate in this field, and for her intense commitment to scientific dissemination and public education. Demano al padrí Marc Güell, professor i crea del Departament de Medicina i Ciències de la Vida, que vagi a buscar la doctoranda i l'acompanyi fins al lloc que ocuparà a l'escenari. Us convido tots a aixecar-vos i a rebre la nostra doctoranda dempeus. Professora Emmanuelle Charpentier, sigueu benvinguda a la Universitat Pompeu Fabra. For Pompeu Fabra University, the awarding of the title Doctor Honoris Causa is the highest distinction we grant to a person in recognition of her or his scientific contribution, university career, or for values that are clearly identified with our institution. It's an immense honor for us that from today and forever, you are part of our faculty of doctors. Professor Emmanuelle Charpentier, welcome to the Pompeu Fabra University.
Si me das a elegir entre tú y la riqueza, con esa grandeza que lleva consigo, hay amor, me quedo contigo. Si me das a elegir entre tú y la gloria, pa' que hable la historia de mí por los siglos, hay amor, me quedo contigo, pues me he enamorado, te quiero y te quiero, y solo deseo y estar a tu lado, soñar con tus ojos, besarte los labios, sentirte en mi brazo, que soy muy feliz. Si me das a elegir entre tú y ese cielo, donde libre es el vuelo para ir a otro nido, hay amor, me quedo contigo. Si me das a elegir entre tú y mi idea, que yo sin ella soy un ser perdido, ay amor, me quejo contigo, pues me he enamorado y te quiero y solo deseo de estar a tu lado, soñar con tus ojos, besarte los labios, sentirte en mi brazo, que soy muy feliz.
A continuació, demanem al professor Marc Güell, padrí de la doctoranda, que es dirigeixi al faristol a fer la lectura de la laudatio.
Bon dia a tothom. Rectora Laia de Nadal, distinguished guest, esteemed faculty, students, friends, fellow scientists. Today is a very special day, a very special occasion, on which we will recognize an individual, I think, whose contributions to science have not only revolutionized our understanding of molecular biology, but also, I think, have opened new avenues for tackling some of the humanity's most pressing challenges. And definitely, this is not a topic. It is an honor to stand before you and give this laudatio speech for Dr. Emmanuelle Charpentier. As we celebrate her receiving this honorary doctorate, recognition befitting a scientist whose work has not only pushed the boundaries of science, but also, of course, had an enormous impact. Dr. Charpentier's groundbreaking research into the fundamentals of CRISPR-Cas9 mark, I think, one of the very few inflection points in biology, not only in the specific field, but also in the broader scope of life sciences. CRISPR developments fundamentally shifted the paradigm of genetic research or molecular sciences, enabling us to address challenges that range from hereditary diseases to the future of agriculture, and all while sparkling vital ethical debates of how far science should go in engineering life. We confer upon Dr. Charpentier this honoris causa doctorate, not only to recognize her exceptional talent and relentless pursuit of knowledge, but also to celebrate her embodiment of the values that lie at the heart of the scientific inquiry, which is curiosity, creativity, collaboration, and ethical responsibility. Her work is a testament to the fact that science, when driven by the power of basic knowledge and basic discovery, can transcend the frontiers that were once beyond our imagination. Emmanuelle Charpentier's journey into the world of science began in Juvisy-sur-Orge, a small town outside of Paris, where she was born. From an early age, Charpentier exhibited an insatiable curiosity about the world around her. This curiosity, nurtured with a supportive environment, led her to pursue an education in science, where she was captivated by the complexities of life at the molecular level. Her passion for biology, combined with her love for problem solving, set the stage for a career that would one day revolutionize life sciences. Charpentier's early career started with undergraduate studies in biochemistry, microbiology, and genetics at the Pierre and Marie Curie University, now part of Sorbonne. These studies contributed to the foundational knowledge that would later serve as a basis of her groundbreaking work. It was during this time that she began to understand the power of molecular biology to answer some of the most fundamental questions of life, questions that would guide her research for the years to come. After completing the undergraduate degree, Charpentier embarked on a master and a PhD program in microbiology at the Institut Pasteur in Paris, where she conducted research on bacterial mobile elements and antibiotic resistance in the laboratory of Patrice Courbalin. It was here where she began to explore the world of molecular mechanisms in bacteria, an area that would later probably prove rather important in her more well-known work. In her postdoctoral years, Charpentier's academic journey took her across Europe to the United States, where she worked in some of the most prestigious universities, including the Rockefeller University, St. Jude Children's Hospital in Memphis. And this experience allowed her to broaden her scientific horizons, exposing her to the field, to different fields and perspectives. In 2022, she returned to Europe and held faculty positions in the University of Vienna, in Umea University, the Helmholtz Center of Infection and Research in Braunschweig, and now she's the founder and director of the Max Planck Unit for the Science of Pathogens in Berlin. As she moved from institution to institution, her commitment to solve the mysteries of molecular biology continued, and as you know, they very, very especially flourished. Despite the many challenges that come with building a scientific career in the highly competitive world of scientific research, Charpentier really continued to pursue of knowledge, and this exploration of basic science built the foundations of her historic breakthrough with CRISPR. Professor Emmanuelle Charpentier focused primarily on the molecular mechanisms of pathogenic bacteria, particularly Streptococcus pyogenes. Indeed, both bacteria and viruses have been extremely prolific in providing us with the most essential tools that we use in the field of life sciences, like thermostable polymerases, ligases, reverse transcriptases, you name it, which greatly advanced life sciences and biotechnology. She investigated how these bacteria regulate gene expression through RNA molecules, specifically studying small regulatory RNAs that play a pivotal role in bacterial virulence and immune evasion. Her work was centered in understanding the bacterial RNA processing and regulatory pathways, which laid the foundation for her later groundbreaking characterization and discovery of the CRISPR-Cas9 machinery. Essentially, CRISPR is a natural defense mechanism used by bacteria to protect themselves from viruses. When a virus attacks the bacteria captures a small piece of the virus DNA and stores in its own genome. So then, if the virus attacks again, the bacteria has this memory and can basically recognize and remember this pathogen. So then it's kind of really an adaptive immune system. While studying S. pyogenes, Charpentier and her team uncovered key insights into how the CRISPR system, originally a bacterial immune system, could be harnessed as a powerful tool for precise genome targeting. Her discovery of the interaction between the Cas9 protein and the guide RNA was pivotal in this development of CRISPR-Cas9 as a really revolutionary technology for targeted gene modification. These contributions are really nothing short of revolutionary, which not only have transformed the field of genome editing, but also opened the door to countless new applications. The discovery and characterization of CRISPR, which stands with this quite long development of clustered, regularly interspaced short palindromic repeats, represents one of the most significant scientific breakthroughs in the 21st century. CRISPR does something very, very basic, but profoundly important. Many of us are familiar with Microsoft Word. Think of CRISPR as the search tool. The Cas9 protein acts like a Control-F command, and then the gRNA, with the guide RNA, is the specific text that we put in the search box. After finding the target system, the CRISPR-Cas9 system works by introducing a double stranded break at the DNA at the precise location. Then this can be repaired in a way that allows scientists to either remove or add or edit genetic material. This level and programmability and precision was unprecedented in the field of genetic engineering, where previous methods were often much more labor-intensive, and CRISPR changed all of that, providing a simple, cost-effective, and highly accurate method for editing genes. This incredible demonstration that the CRISPR system could be adapted as a function for a programmable tool for cutting DNA at the precise locations in any genome. Some work that also the Lithuanian professor Vyšnis Signis had also been working on, and later that year also published in 2012. CRISPR is a very special technology, and Streptococcus pyogenes Cas9, a very special CRISPR system. Actually, I remember hearing one of your interviews that this project was especially progressing quite smoothly and efficiently, delivering the expected results, something that not always happens in life sciences. This reminds me of beautiful symmetry in science during the time that I was doing my postdoc in Boston. So Prashant Mali, a very talented fellow postdoc in the Church Lab, came across one of your publications in 2012, and immediately he recognized the vast potential of this technology, and he very quickly started applying the CRISPR system in human cells, and in a remarkable short time, the results were really astounding. I had the privilege to help him to analyze this data, and I remember how we could hardly basically believe that the results were true, because they were so impressive. And this was published. And I think this kind of Prashant success also truly underscores how quickly and how profoundly CRISPR could be adapted to more complex biological systems I think, further highlighting the groundbreaking work, the groundbreaking power of your discovery. The implications of this technology are vast and far-reaching. In medicine, CRISPR holds the potential to treat a wide variety of diseases, including genetic disease, immuno-oncology, metabolic disease, by editing these defective or creating new alleles that could have therapeutic potential. Clinical trials are already underway. Some CRISPR medicines already have very impressively been approved. And for sure, this will increase the number of therapies that basically can help patients suffering with previously untreatable diseases. CRISPR has been fundamental in expanding our horizon of genetic engineering. Today, our sites, or our possibilities to make, for example, xenotransplantation a reality, a topic very close to my heart that I worked several years in my past, that it was for a very long time a dream. For example, I remember that we always had to hear a joke when working in xenotransplantation, let's say 10 years ago. Xenotransplantation is the future, and they were saying it will always be. Which was very unpleasant to hear. However, largely thanks to CRISPR, this joke cannot be done anymore. Earlier this year, a pig with more than 50 CRISPR edits was used as a kidney donor for a patient in need. These pigs probably are the most sophisticated biotechnological products on Earth, and clearly CRISPR was completely fundamental to make this possible. This is just one example. But the impact of CRISPR is not limited to medicine. In agriculture, CRISPR is being used to develop crops that are more resistant to pests, diseases, and environmental stresses, which could yield to higher crop yields, greater food security, and especially in regions that are more vulnerable because of the effects of climate change. CRISPR is also, of course, being explored as a tool for controlling the spread of invasive species and even for the conservation of endangered species, offering new hope for preserving biodiversity in the face of environmental degradation. I think also very important, our enhanced capacity to engineer biological systems is being amplified by the AI, artificial intelligence revolution. I think here technologies like CRISPR are fundamental to unleash all of this potential. AI transforms data into knowledge, but then mapping the intricate relationships of our genome, predicting the effects of the modifications. Of course, we need tools to really basically edit and implement all these new advancements. But of course, all this knowledge is not enough without a tool that we can precisely translate these predictions of these new systems of AI into reality. CRISPR has a very important role in bridging the gap by providing an adaptable and precise method for editing genomes, turning the predictions made by AI into tangible outcomes, whether it's correcting genetic mutations, enhancing agriculture resilience, or engineering microorganisms for industrial applications. I have the feeling that, well, actually the decades following physics revolution of quantum theory, if we think a little bit from an historical point of view, right, and relativity, this was the decades after these big milestones of physics were very prolific for society, right? We had giving rise to transformative technologies such as atomic energy, lasers, radars, transistors. Today, I think we are witnessing a similar revolution in biology, where biology is really exiting the lab, and the most sophisticated, and biology is the most sophisticated and unique matter in the known universe. And of course, the ability to edit life is fundamental. In its fundamental language, DNA, of course, is completely essential to transform all these dreams into realities. This capability is already transforming, and it will continue to revolutionize our approach to solve some of humanity's most pressing challenges. Much like Leonardo da Vinci, who connected his insatiable curiosity with groundbreaking innovations, the key of future progress lies in bridging this basic scientific understanding with practical applications. By integrating our growing knowledge of biology into real-world solutions. We stand on the brink of the unprecedented advancements that could reshape the future of humanity. I once heard David Walt, founder of Illumina and faculty at the Wyss Institute, at Harvard, making a very interesting reflection. He really liked to measure the scientist not in outputs, more in outcomes. What do I mean by that? So being outputs, how many papers, how many patents. Outcomes being how many people read your papers, how many people read your patents. Yeah, definitely Dr. Charpentier has been cited more than 50,000 times, I think, last time I checked, and her patents have been licensed to companies that raised hundreds, if not millions of dollars. I also heard my previous boss, George Hart, saying that the word impossible was forbidden in the lab meetings. Of course, how can we change the scientific paradigms without looking beyond the horizon? Dr. Charpentier's work has not only advanced the field of genetics, but also has had this very profound impact on society at large. The development of CRISPR has sparked this global conversation about the ethical implications of genome editing, raising very important questions about how we should use this powerful technology. Should we use to modify the human germline, making changes that will be passed down to generations? How do we balance the potential benefits of genome editing with the risk of unintended consequences? Charpentier has always emphasized the importance of responsible research and the ethical considerations in the use of genome editing technology. She has called for the international collaboration and the development of regulatory frameworks to ensure that CRISPR is used in ways that are safe, equitable, and beneficial to society. Her leadership in this area has helped to shape the global discourse on the responsible use of genome editing technology, ensuring that the scientific progress is balanced with ethical responsibility. The societal impact of Charpentier's work extends beyond the scientific and ethical dimensions. Her success as a female scientist is, of course, a role model. I believe that the 2020 Nobel Award in Chemistry was the first awarded to female scientists. Beyond the achievements of Dr. Charpentier has made significant contributions to the future of science through her mentorship and leadership as the director and founder of the Max Planck unit of the science of pathogens. Charpentier has fostered a research environment that emphasizes collaboration, curiosity, and creativity. She has mentored numerous young scientists, encouraging them to pursue bold, innovative research that challenges the status quo. Her guidance has helped to shape the careers of future leaders in the field of molecular biology and biotechnology, ensuring that her legacy will continue through the work of the next generation. It was actually quite beautiful to see that in all of your interviews, the first people that you were thanking were your students, mentees, which it reflects the deep appreciation and collaborators, which it reflects your deep appreciation of the collective effort behind the scientific discovery and your commitment to nurturing this next generation of researchers. You consistently highlight the importance of bringing new scientists to the fundamental sciences to continue fueling the pursuit of knowledge. Your emphasis on collaboration and mentorship underscores the idea that science is not a solitary pursuit, but a shared endeavor that thrives on diverse perspective and exchange ideas. Dedication to cultivating a young mind is as vital as progress as the research itself. You have long advocated about the importance of fundamental microbiology, emphasizing how essential it is to our understanding of life and disease. It is indeed an interesting irony, or perhaps some form of divine justice. In the middle of the 2020 pandemic, a researcher focused on microbial pathogens got the Nobel Prize in Chemistry, in a time when the world was battling a global viral outbreak. The recognition on your work on, at the end, Streptococcus pyogenes and CRISPR highlights how critical the study of microbes is for tackling the challenges that face humanity. And it's a powerful reminder that the answers of some of our most pressing societal problems often lie in the deep exploration of the smallest organisms. As we look into the future, it is clear that Dr. Charpentier's work will continue to influence the trajectory of science and medicine for the decades to come. While CRISPR technology is already transforming fields like genetic medicine, agriculture, and conservation, we are only at the beginning of this revolution. As researchers continue to refine CRISPR and develop new applications, we may one day be able to cure more and more previously untreatable diseases, and even combat the impacts of climate change through genome editing. Charpentier herself has expressed her belief that the full potential of CRISPR is yet to be realized. She has pointed to the possibility that developing more precise genome editing tools, which could further reduce the risk of off-target events, can occur during the editing process. Additionally, she has advocated for the responsible exploration of gene editing in human germline cells, where the changes would be passed down to future generations, stressing that such technologies must be approached with caution, care, and deep ethical consideration. Emmanuelle Charpentier is not an inspirational figure only for women in science, but also for being also a truly international scientist. She also draws inspiration from both art and science, suggesting that the creative methodologies, emotions, and inspiration found in arts parallels the discovery process in research. Specifically, something that I also have heard you highlight, that the methodology is important, both in arts and sciences. You have to know the basics, but master the methods, which requires persistence, but also combine them with creativity. And this combination of creativity and persistence is important, very important for biological research. Also, Charpentier upholds that scientists should think like entrepreneurs, encouraging them to take risks, explore unknown paths, and pursue big ideas with persistence. Her vision empowered scientists to lead and innovate across disciplines, combining scientific rigor with creative inspiration. In closing, we are here to celebrate this truly extraordinary individual whose contributions have changed the course of scientific history. Emmanuelle Charpentier's work in CRISPR-Cas9 has opened new horizons for life sciences, medicine, and countless other fields. Her discoveries have not only accelerated life sciences, but have also given us the tools to shape the future in ways that were once quite unimaginable. But perhaps more importantly, Charpentier has shown us that science is not just about solving problems, it's about asking the right questions. Her work reminds us that the pursuit of knowledge is a continuous journey, and that requires not just intellect, but curiosity and perseverance. Through her dedication to responsible research, ethical leadership, and mentorship, she has inspired the generation of scientists that has set a new standard for what it means to be a leader in the scientific community. Dr. Charpentier, we are deeply grateful for your contributions, and with the utmost respect and admiration, we confer upon you the title of Doctor Honoris Causa. May your journey continue to inspire us all, and may your science guide us in the years ahead. I'm glad that you did not follow the example of Jean-Paul Sartre and of not accepting the Nobel Prize for fear that it would affect his writing. I think this 2020 Nobel Prize has been huge for the field. I would say for life sciences in general. And for contributing to generate such a role model for young and not so young scientists. And thank you, and my most sincere congratulations.
Demanem a Laia de Nadal, rectora de la Universitat Pompeu Fabra, que pugi a l'escenari per tal de procedir a la investidura.
So by the Board of Governors of Pompeu Fabra University, you have been nominated Doctor Honoris Causa in testimony and recognition of your relevant merits. By virtue of the authority that has been conferred on me, I'm handing over to you this medal, a symbol of this title and of the supporting diploma. As you have been admitted to this university, receive in the name of the University Senate an embrace of fraternity from those who are honored and pleased to be your brothers, sisters, and colleagues. That's for you. Thank you. Thank you. amb el diploma. Sorry. Thank you.
Hello, everyone. It is very heavy, actually. So, dear Rector, Dr. Laia de Nadal, dear Vice Rector for Research, Dr. Cristina Pujades-Corbi, dear Barcelona Biomedical Research Park Director, Dr. Jordi Camí, dear Catalan Minister of Research and Universities, Núria Montserrat, dear President of Board of Trustees, Montserrat Vendrell, dear Professor José García Montalvo, dear Director Jordi García Ojalvo, Vice Director Berta Alsina, Professor Marc Guel, and all members of the Department of Medicine and Life Sciences, distinguished faculty, professors, researchers, and colleagues, dear students, graduates, undergraduates, dear ladies and gentlemen. So first, thank you very much for the very kind introduction. What a privilege and honor it is for me to receive an honorary doctorate in medicine and life sciences from your university, Universitat Pompeu Fabra in Barcelona, with a French accent, a European leader in research-oriented education, and it's a pleasure to celebrate it with you. Research is a collective work, and this recognition would not have been possible without the hard work and dedication of a team of young researchers with whom I have worked in a very cheerful, enjoyable, and dynamic environment for the incredible discovery that is a CRISPR-Cas9 mechanism and derived technology. That's why this recognition also goes to my former colleagues and team members, and I am happy to share it with them. To name just a few of the younger team members who contributed to the work rewarded today: Maria Eckert, Eliza Delcheva, Christoph Schelinski, and Martin Jinek. I would also like to thank my colleagues who joined me on the CRISPR-Cas9 journey and trusted my advice, hypotheses, ideas, and interpretations, visions, strategies, plans, and wishes in this project, which I carried out mainly in Austria, Sweden, and then Germany. My personal achievements, recognized by this award, would not have been possible without the institutions that enabled me to establish my laboratories and develop my research projects, and without the funding agencies in Austria and Sweden that generously provided financial support for my project ideas. My special thanks go to the MIMS, the Laboratory for Molecular Infection Medicine Sweden, at Umeå University in Sweden. That is a Nordic part of the Nordic EMBL partnership in molecular medicine, where I spent so many days and nights dissecting, planning, and directing the CRISPR project and discovery over dark winters and long, bright summers, because it's very, very up north. This supreme distinction mainly rewards my work and that of my team and collaborators on the transformative CRISPR-Cas9 gene editing technology. This technology, commonly known as programmable genetic scissors or the Swiss Army knife of DNA, enables the precise modification of DNA, genes, and genomes in living organisms. Let's consider the genome, coding and non-coding, as a text, series of genes as sentences, genes as words made of letters that define the code of life, the basis of DNA. The CRISPR-Cas technology can search for a gene or a piece of DNA and modify the gene, or more precisely, the code of interest at will, just as a software program will search for a sentence or word or a letter in a text to modify, correct, delete, or add a word or letters. That is the CRISPR-Cas technology that makes it possible to rewrite the code of life in a precise, versatile, and efficient way. Soon after the publication of our work in Nature in 2011 and Science in 2012, CRISPR-Cas9 became in record time a popular and versatile genetic tool for modifying genes and their expression in all types of cells and organisms, from bacteria and plants to animal and human cells. Considered one of the most transformative breakthroughs in the life sciences, CRISPR-Cas9 has opened an unprecedented possibilities for medicine, agriculture, and many other fields, and holds great promise for many biotechnological and biomedical applications. Research into genome editing and the use of CRISPR-Cas has indeed revolutionized many areas of the life sciences, from macrobiology, the study of microbes ranging from bacteria to viruses, archaea, yeasts, and fungi, to infection biology, immunology, cell biology, developmental biology, cancer, and brain research, using research models ranging from flies, fish, mice, plants, human cells, and organoids. In the space of just 12 years, numerous scientific papers have been published on the development of the CRISPR-Cas gene editing technology, from version 1.0 to the multiple subsequent versions that have improved the technology's specificity, efficiency, and versatility. Since the discovery of CRISPR-Cas9, a wealth of knowledge has been accumulated on the understanding of molecular and cellular mechanisms in a growing number of cells and organisms. In the agriculture sector, it offers new possibilities for cultivating plant varieties that are safer, better adapted to sustainable production systems, and more resistant to climate change. In the medical sector, gene editing using CRISPR-Cas is making a major contribution to the development of innovative therapies, such as immunotherapy for cancer and gene therapy for hereditary diseases, and also metabolic disorders, and so on and so forth. Numerous screens have been developed in human cells, tissues, and organoids, and many editing human cells, tissues, and organoids have been generated with the aim of better understanding disease mechanisms, finding new targets for therapies, and developing more clinically relevant disease models for testing drugs and medicines in development, greatly benefiting research and development in the pharmaceutical industries, biotech companies, laboratories, and clinical and academic institutions. A first concrete example of a CRISPR-Cas9-based medicine to treat human genetic diseases is the first approved cell-based gene therapy, Casgevy, which uses the CRISPR-Cas9 gene editing technology to cure sickle cell disease, a blood genetic disorder, and was developed by CRISPR Therapeutics and Vertex Pharmaceuticals. CRISPR Therapeutics, which I co-founded over 10 years ago with Rutger Novak and Sean Fowey. It's a great achievement for Rutger Novak, his teams at CRISPR Therapeutics, and later Vertex Pharmaceutical teams, who joined the effort to develop a cure for patients just 10 years after the discovery of the CRISPR-Cas9 technology. Scientists, developers, and clinicians celebrating their success, which began with an obscure research project in my laboratory aimed at, in particular, at understanding the role of small RNA molecules in a bacterium, Streptococcus pyogenes, that causes disease in humans, and how this bacterium uses CRISPR-Cas9 to defend itself against virus attack. CRISPR-Cas is a perfect example of how fundamental research into what might initially have appeared as an obscure set of repeats in bacterial genomes first evolved into the discovery of an immune system in prokaryotic organisms, bacteria and archaea, and later on into this powerful technology. In my laboratory, we first identified a novel small RNA molecule, tracer RNA, in Streptococcus pyogenes. We then understood that this tracer RNA molecule worked with the CRISPR-Cas9 system to enable Streptococcus pyogenes to defend itself against viral attacks. We deciphered the tracer RNA CRISPR-Cas9 molecular mechanism, our work leading to the exploitation of CRISPR-Cas9 as a powerful technology, the gene scissors. Long life, fundamental research, always at the origin of technological, medical, and other innovations. Long life, publicly funded research universities like the Universitat Pompeu Fabra, Barcelona, and institutions that tirelessly support teachers in training the next generation of scientists and researchers in their quest of knowledge and curiosity for a better world. Telling you about my scientific journey could take a whole day, but what I can tell you today is that along the way, many places and people have shaped my life and my research. Mobility has played an essential role in my career and has been a process of self-improvement. Scientific research is a collective endeavor and has become more interactive, multidisciplinary, and multicultural than ever. Scientific research worldwide, including our research that led to the development of the CRISPR-Cas9 genome engineering technology, benefits enormously from international exchanges, whether in the form of young visiting scientists or international collaborations. The value of the mobility and diversity of international researchers goes beyond the benefits to individuals. These exchanges bring fresh ideas, new perspectives, and different ways of approaching problems, all of which are catalysts for the innovation that helps us tackle global challenges such as climate change, cancer, and infectious diseases. Perhaps more subtly, these interactions show us that despite our differences in culture, ethnicity, and nationality, the reasons why we work together far outweigh those that might drive us apart. And in these uncertain post-truth times, when the fundamental values of science are under threat, it is crucial that all countries keep an open door to international scientists, especially those from countries where mobility is necessary. In conclusion, I would like to address the new generations, as I saw some younger people in the back. Science is experiencing unprecedented growth thanks to the development of technologies that are themselves the fruit of discoveries by basic science, which in turn, thanks to the access to so many new innovative technologies, is witnessing an unprecedented number of transformative discoveries every day, a positive feedback loop of innovation between technology and basic science. These are exciting times. Science and technology through innovation are so powerful that they offer and will continue to offer solutions to the challenges we face today. More sustainable agriculture to protect the environment and feed the planet, water for all, health for all, and more peace. We need a new generation of talented, enthusiastic, and ambitious people to tackle the many problems that remain to be solved. Be inspired, be a source of inspiration for those who follow you. Be persistent, tenacious, curious, passionate, and well-informed. Have a plan, develop other plans, because research isn't linear, life isn't linear, and you'll find your plans constantly challenged by unexpected obstacles, but also pleasant surprises. Every challenge you encounter along the way will force you to reflect on yourself. It will broaden your outlook and teach you something new about yourself. That's what makes challenges such a valuable experience. For me, it has been a process of self-improvement. With that, I would like to thank again my colleagues here at the Universitat Pompeu Fabra, Barcelona, for this great honor. Long life to flourishing research in Barcelona. I have had the chance to come back again. I was here more than 10 years ago to visit the EMBL Partnership CRG Institute, and this was actually within the frame of my work at Umeå University, also in EMBL, in an institute that was part of EMBL Partnership, and I had the chance to visit this wonderful site, and I was wondering at the time, why did I choose Umeå? Why Barcelona had also an institute affiliated with EMBL. But at the time I was not known, no one cared about me. But I could really at least appreciate the environment here, and also to see the sea. I guess it must be very inspiring for all of you. But I hope to come back, and as I was invited this morning to give a lecture to the students and postdocs to tell you how we came in details to the CRISPR-Cas9 story. So again, it's really a privilege for me to be here. I'm very honored. CRISPR-Cas9 has brought so many things to my life, and for a scientist, at the end of the day, we do science to be hidden somewhere in our labs with our teams, trying to do our best. And sometimes science can lead someone to very unexpected ways. This has been my case, and I thank you very much for the privilege to be honored today. Thank you.
Je vous parle d'un temps que les moins de 20 ans ne peuvent pas connaître. Montmartre en ce temps-là accrochait ses lilas jusque sous nos fenêtres, et si l'humble garni qui nous servait de nid ne payait pas de mine, c'est là qu'on s'est connus, moi qui criais famine et toi qui posais nu. La bohème, ça voulait dire on est heureux. La bohème, nous ne mangions qu'un jour sur deux. Dans les cafés voisins, nous étions quelques-uns qui attendions la gloire. Et bien que miséreux, avec le ventre creux, nous ne cessions d'y croire. Et quand quelques bistrots, contre un bon repas chaud, nous prenaient une toile, nous récitions des vers groupés autour du poêle en oubliant l'hiver. La bohème, ça voulait dire tu es jolie. La bohème, et nous avions tous du génie. Souvent il m'arrivait devant mon chevalet de passer des nuits blanches, retouchant le dessin de la ligne d'un sein, du galbe d'une hanche, et ce n'est qu'au matin qu'on s'asseyait enfin devant un café crème. Épuisé mais ravi, fallait-il que l'on s'aime et qu'on aime la vie. La bohème, ça voulait dire on a 20 ans. La bohème, et nous vivions de l'air du temps. Quand au hasard des jours, je m'en vais faire un tour à mon ancienne adresse, je ne reconnais plus ni les murs, ni les rues qui ont vu ma jeunesse en haut d'un escalier. Je cherche l'atelier dont plus rien ne subsiste. Dans son nouveau décor, Montmartre semble triste et les lilas sont morts. La bohème, on était jeunes, on était fous. La bohème, ça ne veut plus rien dire du tout.
So first of all, thank you very much, Sol Picó. I don't know where is she. Sol Picó is a well-known choreographer and a dancer based in Barcelona, and she's an expert in classical Spanish and contemporary dance, so it has been, I think, an absolute pleasure for us to have had the opportunity to enjoy her splendid performance. Benvinguts, doctora Emmanuelle Charpentier, presidenta Montserrat Vendrell, doctor Marc Güell, rector Josep Joan Moreso, membres de la comunitat universitària, autoritats, amics i amigues. It is an honor and a privilege to have Dr. Emmanuelle Charpentier as part of our community of honorary doctors. And I would like to express my most sincere gratitude to her for accepting this distinction with James, as you know, to recognize her extraordinary contribution to science and society. També vull agrair al professor Marc Güell, l'excel·lent laudatio, que ha posat de manifest tant la trajectòria com l'impacte extraordinari de la recerca de la doctora Charpentier. I precisament, i us avanço que no seré molt original perquè s'han dit moltes coses, primer amb el Marc Güell, després amb la nostra honoris causa, que coincidim, però tot i així crec que val la pena que faci un resum potser del que s'ha dit i també afegint algunes coses més. Per tant, precisament jo el primer que us volia parlar a la meva intervenció és la qualitat de la recerca o sobre la qualitat d'aquesta recerca i el seu potencial per obrir nous camins. La gran contribució a la ciència de la professora Charpentier, com us deia que ja s'ha dit, és identificar juntament amb la doctora Jennifer Doudna la possibilitat d'utilitzar un mecanisme endogen de reparació de l'ADN que va ser descrit en procariotes, en arquees i en bactèries al principi per editar el genoma de qualsevol organisme. De fet, hi ha organismes eucariotes. Aquest mecanisme, de fet, va ser descobert pel doctor Francisco Paco Mojica, estudiant les arquees i les bactèries que hi havia a les salines de Santa Pola, que no estan molt lluny d'aquí. Ell va identificar aquestes seqüències d'ADN que us han explicat, d'origen viral, que estaven integrades dins dels bacteris i dins d'aquestes arquees de manera repetitiva i que de fet aquests microorganismes les utilitzaven com un mecanisme de defensa quan els virus els infectaven. Per tant, es va descobrir les ara famoses seqüències CRISPR i les que ens han portat avui a fer aquest honoris causa. És a dir, Emmanuelle Charpentier el que va fer o el que va ser capaç de preveure és l'extraordinari impacte que tindria copiar aquesta estratègia molt fonamental de defensa d'uns organismes que són molt senzills, copiar-les com a eina per l'edició de qualsevol genoma. És un exemple preciós de com els descobriments en la recerca fonamental poden tenir un impacte enorme en molts camps, entre ells la recerca biomèdica, i un exemple preciós per remarcar la importància de la recerca fonamental com a eina per transformar la societat. Llegia fa uns mesos un article de la biòloga Ruth Lehmann a Nature Cell Biology en què assenyalava com la línia entre la investigació fonamental i la ciència aplicada és cada cop més difusa. En el seu article, Lehmann defensava com la recerca fonamental havia estat crucial per entendre la biologia i la salut humana i que els descobriments en organismes aparentment insignificants havien transformat, de fet, la biologia moderna. Sustentava aquesta idea amb diversos exemples. El primer, els bacteris termòfils, dels quals va purificar l'atac polimerasa, que és un enzim que és essencial per amplificar el DNA per PCR, una metodologia que avui en dia per la pandèmia Covid-19 és coneguda per tothom. També va posar l'exemple de l'aïllament o la purificació de les proteïnes fluorescents de les meduses, que ara s'utilitzen, entre altres coses, com a reporters en els microscopis, i evidentment, per descomptat, va posar l'exemple de les arquees o els bacteris, on es van descobrir les famoses repeticions CRISPR que avui ens ocupen. La seva tesi, que jo comparteixo, és que si volem ser capaços de transferir coneixement, hem d'apostar primer per descobrir. Per tant, la inversió en la recerca fonamental és imprescindible per garantir el progrés científic, per garantir el progrés de la societat, ja que la recerca aplicada no existirà si no tenim nous coneixements bàsics o fonamentals en què basar-nos. Avançant el camí del coneixement i la transferència exigeix també, i s'ha dit també aquí, estendre la mà als altres i deixar-se ajudar pels altres. En altres paraules, la bona recerca és sempre el producte d'un esforç col·lectiu. Per això avui també m'agradaria destacar aquesta capacitat de col·laboració que no entén ni de fronteres, ni de disciplines, ni de polítiques. La complexitat dels reptes als quals ens enfrontem al segle XXI requereix abordar-los de manera multi- i interdisciplinària. El model docent i de recerca de la UPF ha apostat des del principi per aquesta interdisciplinaritat. Com sabeu, la UPF està organitzada en 8 departaments que cobreixen àrees molt àmplies de coneixement i que, per tant, el que pretén és crear sinergies a partir de les nostres fortaleses. És una estratègia que fomentem i, de fet, intentem donar suport a la interacció entre els nostres recercadors i professors, entre la universitat, entre aquests i els de fora, i també afavorim al màxim les col·laboracions internacionals a tots els nivells. I aquests són els valors pels quals aposta la UPF. Una recerca de qualitat i col·laborativa amb capacitat transformadora. I per fer-ho és necessari captar el millor talent, tenir eines i recursos per captar els millors professors i investigadors, perquè són ells qui, amb el suport del PTGAS, que de fet és el veritable múscul de la universitat, faran una recerca excel·lent i, per tant, després podrem ser innovadors. Deixeu-me ara que us parli, que també se n'ha parlat una mica, de l'impacte social i ètic de la ciència. El descobriment de les professores Charpentier i Dauna va revolucionar l'edició genètica, això ja ho hem dit, i va posar sobre la taula de nou un debat que els científics sempre tenim al cap, el debat ètic. Aquest debat s'ha obert en nombroses ocasions, amb la clonació terapèutica, la fertilització in vitro, la utilització de la intel·ligència artificial, etc. I habitualment som els mateixos científics els primers a identificar els riscos i intentar posar marcs d'actuació ètica. M'agradaria enfatitzar, com bé ha fet el Marc Güell, que la professora Charpentier sempre ha expressat de manera clara la necessitat d'establir un marc ètic rigorós entorn a aquesta eina, entorn a l'edició genòmica per CRISPR i amb les implicacions que té pel progrés de la ciència i la millora de la vida de les persones. Aquest també és el meu parer. La comunitat científica s'autoregula, ens autoregulem. Comptem amb grups de treball i fòrums on es debat i es generen pautes d'actuació per garantir el comportament apropiat. Forma part també d'aquesta transparència que les institucions públiques tenim i hem de tenir vers la societat. Això no implica que el mal ús dels descobriments científics no sigui perillós i tenim, malauradament, exemples en el passat com la bomba atòmica, però hem de confiar en el progrés científic i, per descomptat, ser actius i vigilants. Reivindicar la confiança en el progrés científic també passa per entendre que la recerca no acaba quan tenim la sort de fer un descobriment, i dic la sort. En altres paraules, la transmissió del coneixement o divulgació científica no és un afegit de la recerca o la bona ciència, sinó que constitueix un element que les integra. Si ens preguntem per què és tan important la divulgació científica, m'agradaria aportar una visió diferent a l'habitual i una mica vinculada a l'impacte social i ètic que acabo d'esmentar. Molts dels nous reptes socials que tenim provenen d'avenços científics com poden ser la clonació terapèutica, les teràpies cel·lulars o genètica o la privacitat de les dades, entre altres. També sabem que aquests avenços científics obren reptes, debats i qüestions legals que la llei del moment no pot adreçar. Però en el meu parer, qui hauria de prendre les decisions sobre aquestes matèries no són només els advocats i els científics, sinó que hauria de ser la societat en el seu conjunt amb els governs democràticament escollits. I per poder prendre decisions lliurement s'ha de tenir criteri. En aquest sentit, és imprescindible que des de la societat entenguem que ens hem d'implicar cada vegada més en aquests debats. La ciència és una part present en la nostra vida quotidiana, des de la medicina de precisió fins a les xarxes socials o mòbils. Tot té ciència al darrere. I la divulgació és un instrument, jo diria és l'instrument, que és imprescindible per estar ben informat. Cada vegada més es parla del concepte Science for Policy o ciències per a les polítiques públiques, és a dir, polítiques basades en evidències científiques que ajuden als qui governen a prendre decisions. Un exemple claríssim és el canvi climàtic. Des de fa anys, la comunitat científica ha aportat evidències de l'escalfament del planeta, de la importància de mantenir els ecosistemes, ha alertat del risc de quedar-nos sense aigua. Això són només alguns dels exemples als quals ens enfrontem i que mostren que, malgrat els diferents tempos, normalment entre els polítics i els científics, hauríem de començar a treballar molt més junts i amb la societat, òbviament, al darrere. Parlar del diàleg necessari entre científics i polítics és una manera de defensar el paper central de les persones en el desenvolupament de la bona recerca i el bon ús de la ciència. Però també hauria de ser, i no sempre ho és, una manera de reivindicar el potencial transformador de la recerca feta per dones i el lideratge femení en ciència. Aquest és el darrer punt que m'agradaria tocar en aquest discurs i voldria fer-ho des d'una doble vessant. La primera, de reconeixement a la nostra nova doctora, la professora Charpentier, com a dona que ocupa una posició destacada en el món acadèmic i científic, és un exemple inspirador de lideratge femení. Però abans d'arribar fins on ha arribat i convertir-se en la directora del Max Planck Institute for Infection Biology, aquesta brillant científica ha passat per 25 anys movent-se per 9 institucions científiques repartides en 5 països, un clàssic en molts àmbits científics. Dr. Charpentier, I'm sure that your trajectory, your capacity for mobility and adaptation to new environments and new challenges, so characteristic of the scientific ecosystem, serves as a motivation for many young scientifics who are just starting their academic careers. És possible que el sostre de vidre al qual ens veiem sotmeses les dones, sempre que tractem de promocionar-nos, es trobi avui en unes condicions més favorables que fa 25 anys. Una mica millor sí, però clarament no suficients. I és imprescindible que totes i tots ho entenguem i que actuem. També m'agradaria recordar a totes aquelles figures hidden o amagades, aquelles dones científiques que han estat invisibilitzades i que afortunadament comencen a aflorar. Revisava les dades dels Premis Nobel concedits en totes les categories des de la seva creació el 1901 fins a l'actualitat. En total, 65 premiades versus 912 premiats. És a dir, un 6,7% del total de Premis Nobel s'ha concedit a dones. I continuem veient com s'esvaeixen les oportunitats. La setmana passada l'Acadèmia Nobel ha tornat a fallar en aquest sentit. Cap dona ni en física, ni en química, ni en fisiologia i medicina. Fins quan haurem d'esperar per tal que aquesta situació faci un canvi veritablement? Jo no ho sé del cert, evidentment no tinc la resposta, però acabo ja amb un únic missatge, amb el compromís de la nostra universitat d'apostar per la captació i la retenció de talent també en femení. And to finish, Professor Charpentier, I would like to give you a present of an artist closely linked indeed to science, Joe Milne. Joe Milne was the recipient of an artistic research fellowship from the in-residence program of our Department of Medicine and Life Sciences, and she did a stay among us, exchanging experiences with researchers and looking for synergies between art and science. So the artistic work we would like to give you today is inspired by CRISPR, and we hope you will like it as it combines two of your passions, art and science. So I don't know where is the present. There you go. So Emmanuelle, that's for you. Marc, para tu. Merci beaucoup. Gràcies a tots.
Hem arribat al final de l'acte. Esperem que l'hàgiu gaudit. Moltes gràcies per la vostra presència.